Okay, so hi everyone, my name is Kim Anthony-Gonda. I'm a research and development manager here at Lentigen, and I work with Boro. And together, Boro and I have co-developed the anti-HIV duoCAR-T cell technology that we're going to share with you today.
Yeah, and hi everybody. My name is Boro Dropulić. I'm the Chief Science Officer and General Manager of Lentigen. I'm also the Executive Director of Caring Cross, a nonprofit that's dedicated towards bringing these advanced medicinal cures affordably and accessibly to everyone who needs them. And we're really quite honored to be able to present today. Thanks very much for the organizers for inviting us. What we'll do today is present our work on our anti-HIV duoCAR therapeutic candidate, and also share some thoughts on affordable access of ex vivo and also potentially in vivo approaches. So if I could have the next slide, Kim. So we're going to divide the talk into two parts. Kim will, who really spearheaded this entire effort, will give the first part of the talk, and then I'll give the second part. And Kim will present on a little bit on the background on anti-HIV cell and gene therapy, building on what Chris has already described. Then talk specifically about our anti-HIV duoCAR cells and the results that we've been able to demonstrate, some preclinical animal studies supporting the safety and efficacy of this therapeutic candidate, and then our clinical trial design. Then I'll come back on and share a little bit about the approved CAR-T cell products and their current high cost. Then talk about models for centralized versus decentralized or place of care manufacturing models for CAR-T cells that can dramatically reduce the cost of these therapies. And then also some clinical data showing the promising results of these place of care manufactured CAR-T cells in the lymphoma and leukemia space. And then finally coming back towards optimizing this therapy, a forward-looking slide looking at how you could potentially balance cost with technology feasibility, moving from ex vivo to simplified ex vivo to in vivo, and ensuring safety and efficacy with technology feasibility. So Kim, I'll turn it over to you.
Okay, so the screen looks okay, guys, because I had some issues with it. So it's in slide presentation mode.
You got it.
Great.
Yeah,
okay,
that's great
good.
now.
Okay.
Thanks.
Okay, so as we move towards a cure for HIV, I just want to point out a few landmark studies that kind of help guided the development of duoCAR-T cell therapy for us. And that first landmark study was published in 2002 by Dr. Stephen Deeks and his team at the UCSF. And this was the first clinical study using HIV-specific CAR-T cells to redirect T cells to eliminate HIV. And in this study, he showed that a first-generation anti-HIV CAR-T cell using the CD4 Zeta CAR modality was safe and persisted long-term in people living with HIV. However, this first-generation CAR approach showed limited efficacy. So around that same time, Dr. Dropulić was working on developing HIV lentiviral vectors against HIV. And he showed that HIV vectors encoded an antisense against the HIV envelope gene could inhibit HIV replication, but also protect these protect these CD4 T cells from against HIV. And so this was a second landmark study that, through collaboration with Dr. Carl June at the University of Pennsylvania, they demonstrated the safety of lentiviral vectors for the first time in a human clinical trial. However, this approach also showed limited efficacy in people living with HIV. And so based on this work by these two studies, it was really clear that an integrative approach was needed. So in 2016, I joined Dr. Dropulić at Lentigen, and we began developing these HIV-based lentiviral vectors encoding second-generation CARs that were based on the amazing work of CD19 CAR-T cell therapy, which had shown remarkable success against B-cell malignancies. So in 2019, we published this article showing for the first time a novel second-generation CAR design that we call duoCAR, which can redirect T cells to eliminate HIV, but then also protect those CAR-T cells from infection. And so how does duoCAR-T cells work? Well, duoCAR-T cells consist of two CARs that supercharge the T cells' fight against HIV. And the duoCAR design protects T cells from HIV infection, but also helps to redirect T cells to eliminate cells with HIV that are reactivated from latency. So it's this dual action of duoCAR-T cells that help to reduce the HIV viral loads and prevent loss of CD4 T cells. And the goal of anti-HIV dual-CAR-T cell therapy is to control HIV over the lifespan of the person in the absence of antiretroviral therapy. And so in our in vitro studies, what we saw was that in contrast to mono CAR-T cell therapies, which are based on that older first generation CD4 CAR design, these anti-HIV dual CAR-T cells potently suppress up to 99% or more of HIV infection in human T cell cultures, including some strains that were resistant to broadly neutralizing antibodies currently in the clinic, like VR01. And overall, we found that anti-HIV dual CAR-T cells were highly effective against global HIV subtypes found in America, Europe, Africa, and Asia. To evaluate the efficacy of these dual CAR-T cells, what we do is we inject immune cells with HIV into the spleens of mice, and together with control or anti-HIV CAR-T cells. And then 7 days later, we look at HIV infection levels in the spleens of those mice. And as you can see in these two graphs on the right, dual CAR-T cell therapy significantly suppressed HIV infection in mice, and better than these mono CAR-T cells, which again, remember, they're very similar to these first generation CAR-T cells. And these cells, they can persist, but ultimately they fail to really fully suppress HIV infection and eliminate cells for HIV. And consistent with other groups, we found that the CD4 or single domain CARs designed with the CD4 domain are actually susceptible to HIV infection. But if you look at bispecific CAR-T cells, such as our duoCAR, that has at least one entry inhibitor in combination with CD4 or CD4 CAR, these are actually resistant to HIV infection. And this is recapitulated in mice, where we see if we leave mice untreated or we treat them with unmodified cells, we see CD4 T cells are completely depleted in those mice. However, if we treat those mice with dual CAR-T cell therapy, even after 30 days of HIV infection, we see that CD4 T cells are protected from elimination. And so they are able to survive in these mice. So as we've, over the last year, we've done a number of preclinical studies that have led to an open IND that we're really excited about. And during that process, we've developed a scalable and gentle CAR-T cell manufacturing process that generates what we call slow release CAR-T cells. So these cells are predominantly early memory cells, like stem cell-like and central memory T cells, which, unlike effector T cells, release less cytokines and make a safer and long-lasting CAR-T cell product. So to prove to the FDA that such an early dual CAR-T cell product could be safe and efficacious in treating HIV, we infuse these cells into mice, into humanized mice, and then we assess the impact of dual CAR-T cell therapy on viral loads and safety. And so as you can see here on the right, dual CAR-T cell therapy potently suppressed HIV infection by more than 97%. And this is a result of what we believe these dual CAR-T cells being able to traffic from the site of injection, which in this case is the tail vein, actually to the spleens, where they're able to control this infection in the spleen. And that's why we see this really robust and potent inhibition on HIV. And so in all studies to date, we have observed 100% survival rates of our animals, and we've observed no histopathologies in the major organs, which signifies that this therapy, at least in these models, are safe. So these studies collectively have led to initiation of a phase 1/2A study to evaluate the safety and efficacy of dual CAR-T cell therapy. And so as we translate this therapy to people living with HIV, we've joined forces with Dr. Stephen Deeks, who was the first to conduct an HIV CAR trial in the early 2000s. And so this trial will be the first in-human phase 1 study to evaluate dual CAR-T cell therapy for HIV treatment. And so I'll go just a little bit through the study design. And one thing to note is that for each trial participant, an autologous dual CAR-T cell product will be manufactured and administered on day 0, followed by a treatment interruption. And so this trial consists of 3 cohorts, which has 3 participants each. And so in the first cohort, let's put this on. In this first cohort, you can see that these participants will receive a low dose of dual CAR-T cell therapy, and they won't receive a lymphodepletion strategy. And the second cohort, they will again receive a low dose of CAR T cell therapy, but they will receive a low dose lymphodepletion strategy. And as Chris mentioned, this is just to allow those CAR T cells to engraft and then get rid of some cells so there's space for those CAR T cells. And then in cohort 3, we'll give a higher dose of CAR T cells, one that we know is efficacious from our CD19 CAR T cell therapies or clinical trials. And we'll couple that again with a lymphodepletion strategy. And so these participants will be followed for up to 15 years. And the primary objective of the study is to determine the safety and efficacy of our therapy, followed by analyses of how well CAR T cells persist in people living with HIV, and also the impact of our therapy on the HIV reservoir in certain lymphoid tissues. So with that, I'm going to turn it back over to Boro.
So CAR T cell therapy is curative for many patients suffering from leukemia and lymphoma. In fact, we have 4 approved therapies, as you can see there, for different leukemias and lymphomas. And on the left there, you'll see the ex vivo approach, taking cells from patient, adding the vector to the cells. The cells are genetically modified and then are reinfused back into the patient. And there are many other, even though we have 4 approved products, there are many others in clinical development for a variety of diseases. Next slide, please. So the pharmaceutical and biotechnology companies are pricing these medicines that are not sustainable. The price of each product is at least $350,000 per patient. That doesn't include clinical costs. And this pricing is really not sustainable over the long term for insurance companies or healthcare systems, let alone thinking about the developing world. And so such pricing will restrict access, particularly for the uninsured and marginalized. And biotech, because they have a fiduciary responsibility to maximize shareholder value, it's very difficult for them to price it any lower because they have to satisfy quarter-to-quarter demands by Wall Street. Next slide, please. And so on the left there, you'll see the conventional way that these CAR T cells are made currently. It is a central manufacturing method. And on the right, using technology, you can develop place of care manufacturing at or close to site for a dramatically reduced cost. So on the left there, you'll see the hospitals there in blue. The cells are taken from the hospitals, generally frozen, shipped to a centralized manufacturing facility. The cells are thawed, manufactured, frozen again, shipped back to the hospital, thawed again, and infused back into the patient. And you can see Chimera, a very high cost there. While when you use automation, either within the hospital or within the distance that you can do fresh in and fresh out manufacturing, it's for a fraction of the cost. You can be made, and I know the numbers, for 1/10 of the cost, and I think we can do much better than that. Next slide. And so this is some of the successes that we're seeing with place of care manufacturing of CAR T cells with patients with leukemia and lymphoma. On the left picture there, you'll see our collaborators at Case Western Reserve University. The machine that is to the far left is the Prodigy. That's the closed system machine that's used to manufacture the CAR T cells. And they've had very great success. You can see this on the right there, Case Western Reserve University, an 82% overall response rate. Most of them are complete remissions and low toxicity compared to current CAR T therapy. And to demonstrate the robustness, we also have a site in Moscow in the Russian Federation. And I had the opportunity to visit Michael Mashan, my collaborator, and 2 of the patients there. Again, very high overall response rates, over 90%. And then on the bottom there, our colleagues at the Medical College of Wisconsin, in a case where we only used fresh cells, 12 out of 12 patients had a complete or partial response, 100% overall response rate. So really amazing results using place of care manufacturing of CAR T cells. Next slide. And the reason for it is these current CAR T cell therapies have improved significantly over the technology that was there when the products were approved. And the reason is that now the CAR T cells are predominantly of this T stem cell memory and central memory phenotype. They're not predominantly T effector cells. And when you have less effector cells in the final product, you have less cytokines, and then a lower propensity for cytokine release syndrome or toxicities. So you can think of this, as Kim mentioned, as a slow release CAR T cell product, having a small number of effector cells to do the job, but then keeping a lot of cells in reserve to lower this cytokine production, but also to have more durable responses. And the other thing that we've learned is understanding the CAR-T cell dose. When you have very high tumor burden and a high antigen load and a high T effector dose, well, you get high levels of cytokines and then you run into problems. But by lowering the CAR-T cell dose, you have lower cytokines and then you have good outcomes, but lower toxicity. And you can see there on the right bottom is elimination of a tumor in a 69-year-old patient with follicular lymphoma. So before is on the left and the bright spots is the tumor, and on the right is after the CAR-T cell therapy. Really amazing results. These are very difficult tumors to eradicate. And so for our anti-HIV duoCAR-T cells in our clinical trial, we're going to use a low dose of T cells in the first 2 cohorts, and then just a typical dose in the next cohort. Low antigen load after treatment interruption, because the amount of virus that's coming back after treatment interruption is low. We don't expect to see a lot of activated cells. In fact, we're being very conservative in that regard. And most of these duoCAR-T cells are of the stem cell and central memory phenotype as well. So we think that we've done a very good job in minimizing the risk for any toxicity, but nevertheless, if something would happen, we have the infrastructure in place to deal with any CRS or any toxicity that we would see, because our clinical trial is done in close collaboration with the members of the oncology team as well at UCSF. Next slide, please. So me and my team have been working diligently to generate this global network of place of care manufacturing. Been working with over 100 sites over the world so that they buy these devices, they implement clinical trials at their local facilities. If you see it's an open triangle, it's sort of early discussion, getting things on track. And if it's a closed triangle, that means they either have the devices or they've started their clinical trial. And the nice thing about the network, it's an open network. Each site owns their own machine, so they're free to collaborate with anybody. Next slide, please. And so the next steps for the development, we're initiating a phase 1 clinical trial. It's been allowed to proceed by the FDA. Steve Deeks is the PI and our collaborator at UCSF. Our intention is to demonstrate the safety and efficacy. We'll see what the data tells us, and if successful, determine the effective dose, understand the anti-HIV effects, then develop a place of care manufacturing regulatory approval pathway. We're doing that in collaboration with IQVIA and the California Institute for Regenerative Medicine. Then we want to investigate shorter manufacturing protocols, look into direct in vivo approaches in order to decrease the cost and the time to produce these products. And we're doing that in collaboration with CIRM and the Bill and Melinda Gates Foundation. And then we're also part of a working group called the Global Gene Therapy Initiative, which aims to implement clinical trials for sickle cell disease, beta thalassemia, and HIV in Africa and India. Next slide, please. So this is working towards that balance, if you like, between what we know now works. On the left is ex vivo therapy, and moving in a matrix-like manner to try to develop shorter processes and in vivo approaches to therapy. And the way I sort of see this is sort of a continuum, where you see on the left is high doability, we know it works, higher cost of the product, and working towards in vivo approaches, but then taking on some technology feasibility risk. And what we really need to do as a field is let nature and science dictate the way, work in all these areas in parallel, and then find out that optimal combination of either short ex vivo or direct in vivo with targeted vectors, whatever that is, given safety, efficacy of the product, and of course, most optimal in terms of cost. Next slide. So just in summary, as Kim had mentioned, we had developed a new type of CAR-T cell therapy called duoCAR therapy, which increases the potency of these cells. These cells were shown to be able to reduce, eliminate infected cells, both in vitro, broad strains, and in animal models. And we are developing a place of care manufacturing is feasible for a fraction of the current cost. And these CAR therapies are significantly improved in their safety and efficacy profile over time. And a phase 1 clinical trial evaluating these anti-HIV duoCAR therapy candidates will begin at UCSF shortly. And we're working towards a cure that balances cost, feasibility, and of course, safety. Next slide, please. So as you appreciate, such studies, it takes a village to do such studies. And I really want to particularly thank our patients on our clinical trials and their families. For me, they're our true heroes. And then our collaborators, which are too numerous to list individually. I'll just name them by institution, but we are certainly honored to be working with each and every one of these institutions. Next slide, please. And we're very honored to be here to present our work, and we welcome the community's questions and comments and input. If you want to find out more about Caring Cross, the nonprofit, just look at the website right there, and please feel free to reach out to me at that email address if you have any additional comments or questions after this session. Thank you very much.